May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Luknete, Dr. J.D. Pawar, College of Pharmacy, Kalwan, Nasik. I welcome all of you in this new chapter, that is thyroid and antithyroid agent. Learning outcome of this topic. After completion of this topic, student able to understand the biosynthesis and metabolism of thyroid hormone, draw the structure with IUPAC of drug used in hyperthyroidism with mechanism of action, understand the SAR of drug used in hypothyroidism. So let us start. First one is hormone of thyroid gland. Thyroid hormone are iodinated amino acid derived from L-tyrosine synthesized in thyroid gland and stored as a amino acid residue of thyroglobulin. The thyroid hormone, the tetraiodO L-thyronine and triiodO L-thyronine, play numerous role in regulating metabolism, growth and development and maintenance of homeostasis. The first biologically active iodine containing compound of thyroglobulin was isolated from thyroid extract and named L-thyroxine, or called as T4. This one, T4, L-thyroxine, or called as T4. Later, its structure was established as 3,5-5-tetraiodO L-thyronine. So it is IUPAC name, you can say that 3,5-5-tetraiodO L-thyronine. So here numbering is given. So this ring called as inner ring. So here 1, 2, 3, 4, 5, 6, and here there is a 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. So here there are 4 iodine attached on 3rd, 5th and 3 dash and 5 dash position. Therefore tetraiodO L-thyronine. After 25 years, another thyroid hormone was characterized and identified as 3,5-3-triiodO L-thyronine, normally called as T3. It is the second hormone, T3. IUPAC name here 3,5-3-triiodO L-thyronine. So here there is a 1 less iodine. Here there are 3 iodine on position 3, 5 and 3 dash position. The thyroid gland also containing 2 important iodinated amino acid, that is diiodO L-tyrosine, in short DIT. So it is a DIT. Full form of DIT is diiodO tyrosine, and monoiodO tyrosine, that is MIT. So apart from these 2 main hormone, thyroid gland also contain these 2, MIT and DIT. In addition, there are a small amount of other iodothyronine such as 3,3-diiodothyronine called as T2, this one, T2, and 3,3-5-triiodothyronine called as reverse T3, this one. So the main hormone is T3 and T4. Apart from that, thyroid gland also contain another type of amino acid, that is MIT, monoiodotyrosine, and DIT, diiodotyrosine, and T2 and reverse T3. Then next point, the important role of this thyroid hormone. This hormone is necessary for oxygen consumption, heat production and metabolism of carbohydrate, fats and proteins. It also necessary for proper functioning of gastrointestinal, cardiovascular, reproductive, skeleton, and neuromuscular system. Also important for optimal functioning of catecholamine, antidiuretic hormone, and glucocorticoids, and for normal growth and differentiation. So it is a main important function of thyroid gland. Now next point is that is biosynthesis of thyroid hormone, that is T3 and T4. So it involves the T3 and T4 are formed in thyroglobulin. The major component of thyroglobulin, or the major component of thyroid gland, that is thyroglobulin, is an iodinated glycoprotein. The formation of thyroid hormone depend on exogenous supply of iodide. The formation of thyroid hormone involve following sequence or following steps. So there are various steps involved in biosynthesis of thyroid hormone. So in that, first step is uptake of iodide by follicular cell. Second step, activation of iodide, that is oxidation of iodide and formation of iodotyrosine. Third step, coupling of iodotyrosine residue to form T4 and T3. Fourth step, proteolysis and release of thyroid hormone from thyroglobulin. And last step is conversion of T4 to T3. So now let us discuss one by one these steps. So in that, first step, uptake of iodide by follicular cell. So in this step, the active uptake of iodide, that is follicular cell. So the mechanism enabling thyroid gland to concentrate blood iodide against gradient into follicular cell is the iodide pump, which is regulated by TSH, thyroid stimulating hormone. Decreased store of thyroid iodine enhance iodide uptake. Conversely, dietary iodide can reverse this process. It bring ratio of thyroid iodide to serum iodide, that is called as TS ratio, of 20:1 under basal condition, but more than 100:1 in hyperactive gland. The iodide uptake may be blocked by several inorganic ions such as thiocyanate and perchlorate. Because iodide uptake involves concurrent uptake of potassium, it can also be blocked by cardiovascular glycoside that inhibit potassium accumulation. So here the first step, that is uptake of iodide. So this iodine from the diet is reabsorbed or reuptake by the thyroid gland. That is the first step. Normally the ratio, that is thyroid iodide ratio to serum iodide ratio, is 20:1. This ratio means the concentration of iodide in thyroid gland and concentration of iodide in serum normally is 20:1. But in case of disease hyperthyroidism, this ratio is increases up to 100:1, means 100 concentration in thyroid gland and 1 ratio in serum. So it is increases to 100:1. So here for uptake of iodide pump is involved. So this iodide from the dietary source is reabsorbed by the thyroid gland in follicle cell by the iodide pump. So it is the first step. Then second step, oxidation of iodide and forming of iodotyrosine. In this step, iodide is oxidized to active iodine species that in turn iodinates tyrosyl residue of thyroglobulin. The condition necessary for iodination of tyrosyl residue required iodinating species in higher oxidation state than this iodide anion. The iodinated species is thought to hypoiodate as a hypoiodous acid or enzyme-linked hypoiodate complex. The oxidation of iodide to its reactive species is accomplished by thyroid peroxidase enzyme. It is a membrane-bound heme enzyme that utilizes hydrogen peroxide as a oxidant. Although DIT, that is diiodotyrosine, is a major product, some MIT peptide are also produced. So in this step, after uptake of iodide in first step, there is a uptake of iodide by the iodide pump. Then this iodide is converted into highly oxidized form of iodide by the enzyme thyroid peroxidase. So this enzyme thyroid peroxidase, or also called as TPO, short form thyroid peroxidase or TPO, which is an enzyme, heme containing enzyme, which utilizes hydrogen peroxide, and it causes the oxidation of iodide into highly oxidation state anion, that is hypoiodate. So it is a hypoiodate, or called as enzyme linked iodate or hypoiodous acid. So this form of iodine is active form, or called as higher oxidation state form iodine. So in short, in first step there is a uptake of iodine from the diet or blood. Then this iodine in second step is activated by oxidation process, and this iodide is converted into highly oxidative state, that is hypoiodate or enzyme linked iodate, or called as hypoiodous acid. Then this active form of iodate causes iodination of this residue. So this oxidation state causes the oxidation of tyrosyl residue of thyroglobulin. So it is a tyrosyl residue of thyroglobulin. So here this iodide causes iodination of this. So here there is a iodination. So here this iodine is attached. So here there is a iodination of this iodotyrosyl residue of thyroglobulin. So it is a storage form of tyrosine. So this iodide, that is active form iodide, causes the iodination of this tyrosine residue of thyroglobulin like this. Then third step, that is coupling of two DIT residue and one DIT with one MIT residue to produce T4 and T3. It is believed that this reaction are catalyzed by same peroxidase that affect the iodination, and therefore blocked by the compounds such as thiourea, thiuracil, sulfonamide. Then third step, after this iodination, there is a formation of MIT and DIT. So when one molecule of MIT and one molecule of DIT combine or react with each other, then it forms T3. But when two molecules of DIT react, then it forms T4. So in this step, there is a coupling of MIT and DIT. So in third step, there is a coupling of MIT and DIT. When one molecule of MIT and one molecule of DIT coupled, then it forms T3, and when two molecules of DIT coupled, then it forms T4, and the enzyme which catalyzes this coupling reaction is same enzyme, that is thyroid peroxidase enzyme, called as TPO enzyme. So it is the third step. Now fourth step, that is proteolysis of thyroglobulin and release of iodothyronine. The release of thyroid hormone from thyroglobulin begins with resorption of thyroglobulin by endocytosis into follicular cell and its subsequent complete proteolysis by lysosomal digestive enzyme of follicular cell. Thyroglobulin proteolysis yield MIT, DIT, T3 and T4. Although MIT and DIT are formed, they do not leave thyroid but Instead, they are selectively deiodinated to tyrosine. The tyrosine is recycled into new thyroglobulin, and iodide is recycled into hypoiodate for subsequent iodination. T4 and T3 are secreted by the cell into the circulation. So after formation of or after coupling reaction, there is a proteolysis or there is a breakage. So here after proteolysis, we get four molecules. First one is MIT, that is monoiodotyrosine. Second one is DIT, diiodotyrosine. Third molecule is T3. Fourth molecule is T4. So from these four, MIT and DIT is recycled and converted into again thyroid into the blood circulation. So here there is a separation of T4 and there is a T3. So it is a formation of T3 and T4. So in short, in first step, there is a uptake of iodine from the dietary source by the iodide pump. In step 2, this iodide is converted into highly oxidative state called as hypoiodate, enzyme linked or hypoiodous acid by the enzyme thyroid peroxidase, that is TPO, which uses H2O2 hydrogen peroxide for oxidation. Then in third step, there is a iodination. So here this highly active form of iodide causes the iodination of tyrosine residue of thyroglobulin to form MIT and DIT. Then there is a coupling reaction, third step. So here when one molecule of MIT, one molecule of DIT react, then it forms T3. But when two molecule of DIT react, then it forms T4. It is called as coupling reaction, and this reaction is also catalyzed by the same enzyme thyroid peroxidase TPO. Then fourth step, proteolysis of thyroglobulin to release the T3 and T4. So here after proteolysis, four molecule is obtained, that is MIT1, then DIT, then T3 and T4. MIT and DIT is recycled back and T3 and T4 release into the blood. So it is the whole reaction how T3 and T4 is formed. So it is about the biosynthesis of T3 and T4. Now last step, that is conversion of T4 to T3. Although T4 is the major thyroid hormone secreted by thyroid, it is usually considered to be a prohormone because T4 has longer half-life, much higher level of T4 than T3 in circulation. In peripheral tissue, about 33% of T4 secreted about 33% of T4 secreted undergo 5-deiodination to give T3, and another 40% undergo deiodination of inner ring to yield inactive material reverse T3, that is RT3. The deiodination of T4 is a reductive process catalyzed by the group enzyme named iodotyrosine deiodinase, referred as deiodinase and designated by capital D. About 80% of T3 is derived from the T4. So here the reaction is given about the conversion of T4 to T3. So it is a T4 structure. Here the enzyme is called as deiodinase, designated by capital T. So if we see the reaction is a T4. So as name indicate, 5-deiodinase means this enzyme causes deiodination of T4 at the position 5'. So here is a 5' position. This one is 5' position of T4. So this 5'D enzyme remove this iodine and it forms the T3. So here there is only removal of iodine from this position, that is 5' position. It forms the T3. Then here The enzyme is same, but the position is different. That is only 5 position. Here is the 5 dash position, but here there is a 5 position. So therefore this iodine is removed. 5 number iodine is removed. It forms the product called as RT3, reverse T3. So T4 by the enzyme 5-D, that is deiodinase, it forms T3, and deiodination by the enzyme 5-deiodinase, it forms the reverse T3. Then this T3 by the same enzyme 5-D, here again 5-D. So here there is a deiodination on 5th position, then it forms the 3,3T2, or sorry 3,3-T2. Similarly here this RT3 by the enzyme 5-D, so this iodine is removed, again it forms the same product T3,3-T2. So it is a conversion of T4 to T3. So in short, T4 converted into T3 or reverse T3 by the enzyme 5-D and 5-D. Then this T3 and this RT3 converted into same compound 3,3-T2 by the enzyme 5-D and 5-D. So it is a simple reaction, only removal of iodine by the position shown here. So it is called as conversion of T4 to T3. Then the important, that is drug used in hyperthyroidism. So drug used in hyperthyroidism. So hyperthyroidism means there is a more secretion of T3 and T4, that is thyroid hormone when secreted more, then it is called as hyperthyroidism. So here the drug which are used in hyperthyroidism is the thionamide are the most important class of antithyroid compound in clinical practice used in hyperthyroidism. Thionamide means this thionamide. So R C double bond S and this N. So here is C double bond S, thio. Amide means amide. So here is nitrogen, here is R. So thionamide is the class of drug used in hyperthyroidism. These agents are the potent inhibitor of thyroid peroxidase enzyme, that is TPO. So as we seen in biosynthesis, the TPO enzyme is important in biosynthesis of thyroid hormone. So this agent is an inhibitor of thyroid peroxidase enzyme responsible for iodination of tyrosine residue of thyroglobulin and coupling of iodotyrosine residue to form the iodothyronine. The most clinically useful thionamide are thiouracil, which are 5 or 6 member heterocyclic derivative of thiourea, and it includes thiouracil, 6N-propyl 2-thiouracil, that is called as PTU, and thioimidazole, that is called as 1-methyl-1-mercaptoimidazole MMI, a common name of methimazole and carbimazole. So the drug which are used in hyperthyroidism is type of thionamide, general structure. So here if you see, here there is a N, here is a R, here is C double bond S. It is called a thionamide. When this R is replaced by a nitrogen, then it is called as thiouracil. So it is a thiouracil. Here on both sides there is a nitrogen. Here is not nitrogen, here is also nitrogen, and this one is carbon sulfur bond. So it is called a thiouracil. So there are two types of thiouracil, 5-member ring and 6-member ring. In case of 5-member ring, here example is MMI, methyl mercaptoimidazole, that is MMI, and carbimazole. So the example of 5-member thiouracil is MMI, that is methyl mercaptoimidazole and carbimazole. In case of 6-member drug, example is thiouracil, then methyl thiouracil, then propyl thiouracil called as PTU. PTU means propyl thiouracil. So it is an example of 6-member thiouracil compound. So in case of this compound, that is PTU, methyl thiouracil and thiouracil, more active is PTU. So PTU is more active than the methyl thyroxine, which is more active than the thiouracil. The ability of PTU to inhibit the enzyme 5-deiodinases, the peripheral deiodination of T4 to T3, has made PTU the drug of choice in treatment of emergency thyroid storm. Thyroid storm means sudden increase in thyroid hormone. The SAR of 6-member heterocyclic derivative of thiourea is, first point, SAR in case of 6-member ring. The carbon 2 thioketo or thioenol group and unsubstituted N1 position are essential for activity. So here C2 thioketone, so means this group, ketonic thioketone group on second position, and thioenol, so this undergo thioenol tautomerism as shown in this reaction. So this hydrogen shift here, this bond shifted here, it is called as thioenol tautomerism. So this phenomena is also important for activity. Next point, the ketonic group at carbon 4 and presence of alkyl group at carbon 5 and 6 enhance the activity. So keto group at carbon 4, so it is a carbon 4. Ketonic group at carbon 4 and alkyl group at carbon 6. So this one is carbon 6. It enhances the activity. Methimazole has more PTO inhibitory activity and is longer acting than PTU, but in contrast to PTU is not able to inhibit peripheral deiodination of T4 because of presence of methyl group at N1 position. Effort to improve the test and decrease the rate of release of MMI leads to development of carbimazole, which is a prodrug of methimazole. So here is a carbimazole, which is a prodrug of MMI, that is methimazole in case of 5-member ring. Now the PTU and MMI, that is propylthiourine, and methyl mercaptoimidazole, that is MMI, are taken up by thyroid gland and act as a substrate and inhibitor of thyroid peroxidase enzyme. The thiouracil as a potent inhibitor of thyroidal iodination, and it is also suggested that thiouracil such as PTU, which contains sulfhydryl group, would prevent the formation of thyroid peroxidase enzyme and iodine complex when thiouracil iodide ratio was high, and compete with the thyrosyl residue. The thiouracil alter the conformation of thyroglobulin by binding to thyroglobulin. So these drugs simply inhibit the thyroid peroxidase enzyme, which is involved in the biosynthesis of T3 and T4 as we seen in the biosynthesis process. So these drugs act as a substrate or called as false substrate for the TPO and therefore inhibit the synthesis of T3 and T4. So these are the main drugs which are used in hyperthyroidism, that is thiouracil, methyl thyroxine, propylthiouracil, MMI, and carbimazole. So these are the main drugs which are used in hyperthyroidism. Apart from this, there are another drug that is called as aniline derivative. These drugs also interfere with the process catalyzed by the thyroid peroxidase like iodide oxidation and coupling of iodotyrosine. Example, carbutamide, so it is the structure of carbutamide. Sulfaguanidine, it is a sulfaguanidine para-aminosalicylate. So these are the another drug which may use for hyperthyroidism. So these are the main drugs which are used in case of hyperthyroidism. Now the next, that is drug which are used in hypothyroidism. So here there is a called as anti-hypothyroidism, so or called as anti-goiter. So it is also disease due to the less secretion of thyroid hormone. Previously drugs which are used in case of hyperthyroidism means there is a more secretion of thyroid hormone, but here there is a hypothyroidism means there is a less secretion of thyroid hormone, then it is called as hypothyroidism. and these drugs which are used in hypothyroidism is called as anti-hypothyroidism or anti-goiter agent, or called as thyroid hormone analog. So here there is some SAR points. So here there is a natural hormone, that is T4, thyroxine is the structure of T4. It is a general structure of thyroid hormone. So here first we see the SAR of this thyroid hormone. So in that first site, that is aliphatic side chain. Aliphatic side chain means this side chain. It is called as aliphatic side chain. First point, naturally occurring hormone are biosynthesized from L-tyrosine and possess L-alanine side chain. The L isomer of T4 and T3 are more active than the D isomer. So as we seen in biosynthesis, T4 and T3 are biosynthesized from the L-tyrosine. Here is capital L. Capital L means when the amino group is on the left side. So this side chain is an L form. So it is more active than the D isomer, in that there is this NH2 group is on the right side. So L isomer is more potent than the D isomer. Second point, small b, the carboxylate ion and number of atoms connecting it to the ring are more important for the activity than the intact alanine side chain. In carboxylate series, the activity is maximum with 2 carbon between carboxylic group and aromatic ring, but decreases with either shorter formic acid or longer butyric acid. So here carboxylic acid COH is important. So here if there are 2 carbon between carboxylic group and this aromatic ring, then it shows maximum activity. If we decrease this number of carbon atom between acidic group and this aromatic ring, then it is less active. So if we decrease this 2 carbon with 1 carbon, it is called as formic acid derivative, which is less active. Similarly, if we increase the number of carbon atom above the 2 carbon atom, then the activity is also decreases. So in short, the activity is maximum when there is a 2 carbon between carboxylic group and aromatic amino group, but decreases with either shorter formic acid or larger butyric acid derivative. Third point, that is ethylamine side chain analog, means without carboxylic group of T4 and T3 are less active than the corresponding carboxylic acid analog. In short, when we remove the carboxylic group of side chain, compound activities decreases. Then second is alanine bearing ring chain, alanine bearing ring called as inner ring or alpha ring. So the ring to which side chain is attached is called as inner ring or alpha ring, like this. So it is called as inner ring or alpha ring. The second ring is called as outside ring or exo ring or beta ring. So regarding this ring called as inner ring to which side chain is attached. And that first one is removal of both iodine from inner ring of T4 and T3 to form 3-5-T2 or 3-T1 show less activity. So in case of T4 structure, if we remove the iodine from the inner ring, then it forms T-5-T2. Same, if we remove the iodine from this inner ring of T3, then it forms T3-T1 show less activity. So in short, from T4 structure, if we remove iodine from the inner ring, then it forms 3-5-T2, which is less active. Similarly, if we remove iodine from the inner ring of T3 structure, then it gives 3-T1 show less activity. Then small b point, activity is retained if we replace 3 and 5 iodine with bromine. So if we remove this, activity is retained if we replace the 3 and 5 iodine with bromine. So if we replace the iodine of T3 and T4 with bromine, activity remain. Next point, broad range of activity found with halogen free analog indicate that halogen is not essential for activity. So if we remove the iodine from the inner ring with alkyl group, then the activity is maintained, or in short, iodine. For example, here there is a 3-isopropyl 3,5-dimethyl L-thyrronine. So here iodine is replaced by this methyl group. Therefore, it shows the activity. So halogen is not important for activity. So here the example is 3-isopropyl 3,5-dimethyl L-thyrronine, in which iodine is replaced by this methyl group. Next point, D. Substitution of 3 and 5 position by alkyl group larger than methyl group, such as isopropyl and secondary butyl methyl moiety, produce inactive compound. So if we replace this methyl group with larger group, that is isopropyl and secondary butyl, then the compound is inactive. Then bridging atom. So there is a, in case of T3 and T4, there is a oxygen between these two rings. So it is called as bridging atom. So in T3 and T4, oxygen atom is the bridging atom is oxygen between the two rings. So here the biphenyl analog formed by removal of oxygen bridge is inactive. So if we remove this bridge in T3 and T4, here is oxygen between these two rings. But if we remove this oxygen, then the compound is inactive. Similarly, replacement of bridging oxygen by sulfur or by CH2 produce highly active compound. So if we replace this oxygen by sulfur or by CH2, then it gives the highly active compound. Next is phenolic ring, means outer ring. Another, that is outer ring to which hydroxyl group is attached. So this one is this ring. In that first point, unsubstituted parent structure of this series, that is LT2, possess low activity. Second point, substitution of 3' position by hydroxyl group causes decrease in activity because of decrease in lipophilicity. So on this position, if we substitute hydroxyl group on 3' position, then the activity is decrease because compound is more hydrophilic but less lipophilic, because in R3, in T3 and T4 already one hydroxyl group is present, but if we put one extra hydroxyl group on 3' position, then the compound is more hydrophilic, less lipophilic, and the activity decreases. Next point, substitution of alkyl and halogen group at 3' position result in increase in activity. The order of increase in activity for halogen is fluorine is more than chlorine, bromine is more than iodine, and for alkyl group the order is CH3. Sorry, here iodine is more active than the bromine, then bromine is more active than chlorine, then fluorine. Similarly, in case of alkyl substitution, isopropyl group is more active than ethyl than CH3. So the substitution of alkyl and halogen group on this position, it is called as 3' position. So if we put here halogen or alkyl group, activity decreases. The order of activity of halogen is this, that is iodine is more active than bromine, which is more active than chloro and fluoro. Similarly, in case of alkyl group, isopropyl group is more active than the ethyl and methyl. And the last point is the phenyl substitution at T3 3' position decreases the activity. So on this 3' position, if we put the phenyl group, the activity decreases. So it is on the phenolic ring. Then phenolic OH, that is hydroxyl group, which is present on the outer ring of T3 and T4. So if we replace this hydroxyl group of T4 and T3 with amino group, activities decreases. Similarly, if we replace this hydroxyl group with methyl group, again there is a loss in activity. So it is about the SAR of thyroid hormone analog. Now individual drug, thyroxine. So these are the drugs which are used in hypothyroidism. So in that first one is thyroxine. So it is the structure of thyroxine. Here the numbering is given 1, 2, 3. Then here 1, 2, 3. Sorry, 1, 2, 3, 4, 5, 6. Here is 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. IUPAC name 2 amino means this 2. 2 amino. 3 means this 3. 4 means this 4. 4 hydroxy means this 4 hydroxy. 3 dash, 5 dash. This one is 3 dash. This one is 5 dash. Diiodo, so it is iodine, diiodo. Phenoxy means phenyl with oxygen, phenoxy. Then here there is a 3 and 5. Iodine is present, that is 3,5 diiodophenyl, and this one is propionic acid, acid with 3 carbon, propionic acid. Uses: levothyroxine is used to treat an underactive thyroid, that is hyperthyroidism. It replaces or provides more thyroid hormone, which is normally produced by the thyroid gland. Low thyroid hormone level can occur naturally or when the thyroid gland is injured by radiation or medication or removed by surgery. Having enough thyroid hormone is important for maintaining normal mental physical activity. In children, having enough thyroid hormone is important for normal mental and physical development. This medication is also used to treat other types of thyroid disorder such as certain types of goiter or thyroid cancer. So it is the main use of thyroxine. Next is thyrronine. So thyrronine is a deiodinated form of thyroxine. So it is the structure of thyrronine, which is a deiodinated form of thyroxine. IUPAC name same here. Numbering is 1, 2, 3, 1, 2, 3, 4, 5, 1 dash, 2 dash, 3 dash, 4 dash. So 2 amino, same 2 amino, 3 means this 3, 4 means this 4, 4 dash hydroxy, phenoxy. So here is a phenoxy, phenyl with oxygen. Then this phenyl propionic acid. Then the drug which are used in hyperthyroidism, we already studied. First one is propylthiouracil, PTU. So it is the structure of PTU. So here IUPAC, so here numbering is 1, 2, 3, 4, 5, 6. 6 propyl, so it is a 6 position propyl group. 2 sulfonyl, pyrimidine, so it is a pyrimidine. 4 one, it is a 4 ketonic group, 4 one. Uses: PTU inhibit the enzyme thyroid peroxidase, which normally act as a thyroid hormone synthesis by oxidizing the anion iodide to iodine, facilitating iodine's addition to the tyrosine residue on the hormone precursor thyroglobulin. This is the one of the essential steps in formation of thyroxine. So as we studied biosynthesis, TPO, thyroid peroxide enzyme, is involved in the biosynthesis of thyroid hormone. So these drugs simply inhibit that enzyme and prevent the formation of T3 and T4. Next drug used in hyperthyroidism, methimazole. So it is the structure of methimazole. IUPAC: 1 methyl imidazole ring is there, 2 thione. So it is a 2 thione. Methimazole prevents the thyroid gland from the producing too much thyroid hormone. Methimazole is used to treat hyperthyroidism, that is overactive thyroid. It is also used before thyroid surgery or radioactive iodine treatment. So it is about the drug which are used in hyperthyroidism as well as hypothyroidism. So thank you. If you like the video, then please subscribe the channel. Thank you very much.